Now we're going to move on to talk about genetics. And so genetics, when we talk in psychology, we often discuss in terms of either molecular genetics or behavioral genetics. So one of them is looking exactly down at the DNA, and one of them is just looking about how genetic trends would manifest in a population. So first we'll look at molecular genetics. So molecular genetics is really the study of specific traits on the genome, and how we do this is through the investigation of chromosomes and those traits or alleles on the chromosomes. So in case you're not familiar with the term chromosome, just keep in mind that most humans, but not all, will have 46 chromosomes in most of their cells. The exceptions to that are the sperm and ovum or egg cells. And so the sperm or egg cells differ from ovaries and testes, keep in mind. Testes produce sperm and ovaries produce ovums or eggs. And so the sperm and ovum each have 23 chromosomes. What happens is a sperm with 23 chromosomes and an egg with 23 chromosomes, they meet, they fuse into a zygote, and that zygote will now have 46 chromosomes and will produce many cells that have 46 chromosomes in all but their sperm or egg cells. Now, important to understand is how a sperm and how an egg develop is they are randomly an assortment of anything in your body. Whether your body produces egg or sperm or nothing at all, when you produce either a sperm or an egg, basically it's not just taking the chromosomes you got from your mom or not taking the chromosomes you got from your dad, it's a random assortment. And some of those chromosomes might be hybrids. In that way, sperm are not just replicas of one of your grandparents, and eggs are not just replicas of one of the grandparents. There's an infinite number of combinations, which makes this so fascinating. Offspring can be any array of combo of the parent. Now, important to understand that these 46 chromosomes come, in most cases, in pairs. And so these pairs, the pairs here are sort of colorfully dyed just for your enjoyment. But along these chromosomes, they're kind of long little sticky things. There's different bands. You can see there's a blue, dark blue set right underneath the red set here. And it has different bands of light and dark. And those different bands code for traits. And each trait is coded twice. It's coded on both corresponding chromosomes in the pair. And so those bands are the traits or allele that we're talking about. So on each chromosome, those bands are coded twice. So imagine you have a chromosome that codes for toe length, and one chromosome codes for shorter toes, one codes for longer toes, or they both code for the same type of toe. That's where we're seeing how a lot of our traits, both physical and psychological, are coded twice. And so these alleles or traits have two codes, one from each chromosome in each pair. And so these exact codes, the exact alleles or the proteins that represent them, is what we call our genotype. The genotype is the word we talk about in molecular genetics to refer to what is actually going on in the chromosomes. What proteins are there? What proteins are coding for these alleles? What is happening? And you may have two alleles that match. If your two alleles both match for the same toe length, let's say, then you're going to be considered homozygous. Homo meaning the same, zygote referring to what happens when a sperm and egg combine. So this means that within the zygote, the codes are the same. So this is when the two alleles and corresponding chromosomes match. If you have two codes that don't match, perhaps one code on one chromosome you got from one pair codes for long toes and the other one codes for short toes, well, you're going to be considered heterozygous. Hetero meaning different. So different codes within the zygote. And so based on whether you're homozygous or heterozygous, we're going to see this genotype manifest differently. And so your genotype is different from what we call a phenotype. The phenotype is actually how it manifests. When you look at your feet, do you have relatively long toes or relatively short toes? Maybe you have medium length toes. And so that's going to tell us how it manifests. And so if you are homozygous long toes, your phenotype is probably going to be long toes. But if you're heterozygous, your phenotype may be different depending on which of the two non-matching codes is more dominant. And so for that, we're going to discuss dominance. So when we think about dominance, what we're talking about is the allele or the trait on one of the chromosomes has more influence on a phenotype. And the reason why it might have more influence on a phenotype is because it's chemically more potent. The protein that makes it up is just going to be a bit stronger, more potent than the protein that makes up the other alleles that doesn't match. And so there's lots of physical traits in humans that we know are dominant. Some of these are referred to as the Mendelian traits, things like being able to do a hitchhiker's thumb, which I can do, or rolling your tongue, which I can also do, and that one right there. But other things such as having darker eye color, darker skin color, darker hair color, as well as having curly hair, having detached earlobes. So whether or not your earlobe kind of goes straight into your head, or if it kind of dangles first. So dangling earlobes would be considered dominant. And we also know that dimples and chin clefts and freckles, and there's many others that are dominant, of course. And then, so these are the chemicals that are just a bit more potent. There's also chemicals or alleles of traits that are less potent. So the recessive traits are considered to be traits with less influence on a phenotype, meaning they tend to only express if you're homozygous recessive. What happens here is if you have one allele for a dominant trait, one allele for a recessive trait, the recessive one is submissive and doesn't show up. It hides behind the dominant trait. And so you usually need two alleles that match. You usually need to be homozygous recessive for these traits to show up. And so examples of these are things like straight hair, lighter colored eyes, lighter colored hair, and attached earlobes. So typically, in order to see a blonde hair, blue-eyed baby, they need to get two traits for the blonde hair, blue eyes. Of course, it is a lot more complicated than that. We know eye color tends to be mapped on 13 different allele pairs, but just an exaggeration. And then things get a lot more complicated than just dominant and recessive alleles. We tend to also see this phenomena called incomplete dominance. So incomplete dominance is when you're heterozygous, but your phenotype is really blended. It's not showing the dominant one and it's not showing the recessive one. It's showing a mix, but in a blended way. We can often think about this as if you had a flower, and if the flower had one red flower parent, red petals, and the other parent had white petals, and the flower, the offspring, had pink petals. That would be considered incomplete dominance because it's happening in a blended way. So in humans, we tend to see lots of blended phenotypes, such as wavy hair could be a mix of curly and straight hair. We also may see people with a medium skin tone complexion if they had a parent with a darker complexion and a parent with a lighter complexion. And we might also see in eye color hazel eyes, where one parent gave them an allele for green eyes and one parent gave them an allele for brown eyes. They don't have brown or green eyes. They have a bit of a hazel that's a bit of a mix. So that could be incomplete dominance. We might also see this in terms of height. With a tall parent and a short parent, you might have a medium height offspring. There's lots of different incomplete dominance where it happens in a blended way. By blended, we mean nearly all the cells are showing this sort of hybrid, or this sort of mixing of the two different genotypes, two different alleles. And then we have what's called codominance. This is when the offspring has two alleles that are heterozygous, and they both impact the phenotype, but in a non-blended way. So in a non-blended way, what we're talking about here is that in some cells, one allele is dominant, and in other cells, another allele is dominant. A good example of this is in people with heterochromatic eye color. So this could be a person who has one blue eye and one green eye, or one brown eye, one blue eye. So what's happening here is in the iris of one eye, the blue allele is dominant, but in the iris of another eye, the brown allele is dominant. We may also see this in individuals who have one hair color on the top of their head and another hair color on their beard. Tend to see a lot of men of Irish descent with this phenomena, where their hair on the top of their head is brown, but then their beards grow in red. So that could be codominance. Important to note, freckles are not codominance. Freckles are just a phenomena that's considered dominant. But we also might see this in terms of asymmetrical earlobes. One earlobe could be attached, one earlobe could be dangling. We might see some people that have curly hair on one side of their head and other parts of their hair grow straight. There are lots of different examples of codominance. When we're just still talking about molecular genetics and how parental alleles can impact the alleles of offspring, it's important to understand that in psychology, we don't get into dihybrid crosses too much at this point. For the purposes of this class, we'll just leave it off with the Punnett Square. And so if you've never seen a Punnett Square before, I just want to walk you through how this works. Let's say hypothetically, we know it's a lot more complicated than this, but for hypothetical purposes, let's say we know that brown hair color is dominant to almost all other hair colors, other than black. So brown's dominant to red. Red hair color is also dominant to blonde, but is recessive to brown. And blonde hair color is the most recessive, meaning in order for blonde hair color to show up, you need to have two alleles for it. You have to be homozygous recessive to be blonde. So let's assume that there's two parents here, and the father is going to have an allele for brown hair and an allele for blonde hair. Because blonde's recessive to brown and brown's dominant to blonde, his goatee has manifested as brown. Then the mother, her genotype has one allele for red hair, one allele for blonde hair, and her phenotype is going to be red hair. And that's because red is dominant to blonde, blonde is recessive to red. And so what happens here is every time the man produces a sperm, the sperm only has 23 chromosomes, and it only gets one of his two possible alleles. It either gets his brown allele or his blonde allele. So 50% of the time when he produces sperm, it's going to contain the allele for brown hair. And 50% of the time when he produces sperm, it's going to contain the allele for blonde hair. The woman, at 50% of her eggs will contain the allele for red hair, and 50% of her eggs will contain the allele for blonde hair. So what this means is if they were going to start a family together and they were going to produce offspring, we would find that 25% of the time they would produce an offspring with the genotype brown-red. Would have one allele for brown hair, one allele for red hair. And because the brown is dominant, they would have an offspring with brown hair. 25% of the time, they would have an offspring with the allele brown-blonde, meaning the genotype has one allele for brown hair, one allele for blonde hair, and the phenotype of that offspring would likely be brown hair, because brown is dominant. 25% of the time, they would provide the offspring with the genotype red-blonde, or blonde-red, if you will, and that offspring will have the phenotype of red hair. And so red is dominant to blonde. And then 25% of the time, they will both contribute an allele for blonde hair, giving their offspring the genotype blonde-blonde and the phenotype blonde. In this example, we can see there's 4 possible genotypes, different genotypes that their offspring could get. There's brown-red, brown-blonde, red-blonde, and blonde-blonde. 4 different possible genotypes. But there's only 3 possible phenotypes. That is, 50% of their offspring will have brown hair, 25% will have red hair, and 25% will have blonde hair. So that's how we distinguish the differences between genotype and phenotype. Genotype being the codes of the alleles, and phenotype being how it manifests. So this works great if we can track something at the genome level, if we know exactly what allele is going to do that. And there are some psychological phenomena that we track in that way, but there's also many psychological phenomena we can't track through Punnett squares. And these are things that are considered to be polygenic traits. Polygenic traits are things that are not just coded on one pair of alleles or one pair of chromosomes, they're coded throughout the genome. And so these are things that are very complex, such as criminal behavior, personality, autism, depression, intelligence. We have not identified a specific allele that codes for these. And so because of this, we have to look to behavioral genetics and look at how this tracks and how this manifests in behavior. And so how do we look at behavioral genetics? Well, we often look at this through family studies. And what we do in behavioral genetics is we map a psychological trait in a parent, and we map a psychological trait in a child, and we do a large correlation study. Think back to Unit 2 when we talked about correlations. You have to do that in a large sample, not just with one group. So we couldn't just look at this for one set of parents and one offspring. We'd have to look at this over a whole population. So let's assume we are looking at something like intelligence, and we measured intelligence in 200 parent-child pairs. So 200 parents, 200 children, we mapped them on, and we drew a correlation chart. We would get a scatter plot, and we would look at the correlation. Parents' IQ increases, does child IQ also increase? And it does. And so this is where we started off looking at behavioral genetics and looking at family studies, but there's a lot of problems with this methodology. And one of the problems is the nature-nurture dilemma. Yes, parent IQ and child IQ are highly correlated, but is that due to genetics or is it due to environment? Is it due to the parent reading to their child or engaging in activities that would expose them to do well on an IQ test? So we have to tease it apart further than that. So we have a few other behavioral genetic techniques we tend to do. One of them is the adopted studies approach. And so for the adopted studies approach, we would look at the correlation in IQ between a child and their biological parent, but also between the child and their adopted parent. And so in this way, we could see how well correlated the kids and their biological parent, who they didn't share an environment with, and with their adopted parent, who they didn't share genetics with, worked. And we could compare those correlations. And then another type of study we may engage in is looking at the correlation in a trait between identical twins who share both DNA and a prenatal environment. And we compare that with the correlation of a trait in fraternal twins who only share a prenatal environment and not identical DNA. And we compare that to non-twin siblings who do not share a prenatal environment or share DNA, identical DNA. And so what we do here, let's say we look at the intelligence correlation with identical twins, with fraternal twins, and with non-twin siblings. We may find that the correlation between identical twins is much stronger than fraternal twins. And because they both shared a prenatal environment, that can help us cancel out a lot of the nurture side. They grew up at the same time, they're the same age, they might have experienced a lot of the same cultural, historical events. And so because of this, that allows us to identify how much of the overlap, how much of the correlation is due to genetics alone. And so this is one of the most powerful ways we can do this. So twin studies are really powerful. Historically, we've also engaged in what's known as adopted twin studies. In the 1960s, we engaged in the Minnesota twin study. What they examined were identical twins that were adopted into separate households and who had closed adoptions. And they were able to reunite the twins for the purpose of the Minnesota twin study after they had assessed them on a variety of personality and cognitive tasks. And there were some twins that were raised in very different households. Some of them raised in very religious homes or very secular homes, or two different religions, or speaking two different languages, yet they still grew up with the same preference and the same hobbies that were not affiliated with just cohort norms. Now, we don't tend to see siblings adopted into separate households anymore, and we tend to see far less closed adoptions and much more open adoptions. Now, unfortunately, adopted twin studies would not work so well today, but they allowed us in the history of psychology to really appreciate and understand how these behavioral genetics were at play. And this has brought us to the end of Unit 3, Biopsychology. Congratulations for making it to the end of this unit.